So today we're going to give a brief history of microbiology and give you some of the background to how some of the things that we're going to be teaching within this course came about. All right, so kind of starting out, we want to know, what does life actually look like? Now, there was a period of time where there was this belief in a concept called abiogenesis. So abiogenesis essentially means from no life, life. So there was a thought that there didn't need to be life in order to develop life, right? We now know that to not be the truth, but how do we get to that point where we start to understand that concept? Well, we start first with, what does it actually look like? So before we had the tools to observe certain things, there was no concept that those things actually existed. So Van Leeuwenhoek was one of the early individuals that helped us give us some insight into what these things actually look like. Now, who was he? Well, he was in textiles, so he dealt with clothing, and he wanted to ensure that the clothing that he produced was of the highest quality and was of the thread count that he said it was. So he developed these kind of rudimentary microscopes, right, in order to look at the threads that was in his clothes. Um, but what he soon found out is that if he used those same microscopes in order to view other things, he saw things that he, at one point, well, essentially no one at that point had ever seen before. He saw things floating around that looked like they were chasing each other in pond water. So he called these things animalcules. You may have also heard them called beasties, right? Well, we now know these things are microorganisms. What he may have been looking at are some protozoa. He may have been looking at some algae, right? Now, he looked at many, many specimens uh, through this rudimentary microscope. Now, this microscope you probably wouldn't consider a microscope from the ones that you've seen throughout your schooling and just throughout the world, but this was kind of the first rudimentary microscope, essentially what we would consider something like a magnifying glass, where it had a small little lens, right? And the specimen holder that actually held the specimen in place so you can manipulate it without touching it with your hand. You hold it up to the light. So it was a light microscope. It's actually what we call a simple light microscope because it only had one lens, while compound microscopes have multiple lenses. So as he started to see these things, he started to kind of get insight into a new world. This is why he's kind of called the grandfather of microbiology, because he is the one that kind of showed us that there are other objects or other living organisms out there besides what we were used to. Now, the common thought before this was a little bit different. The common thought was that things that cause disease were things like smells or spirits or curses, right? We now know that to not be true. Not all diseases come from microbes, but a lot of disease causing agents are microbes. The microbes are the things that are actually causing disease. And we'll get a little bit into that later within this lecture. So once you start to recognize that you have all these different things that are living, well, the obvious thought is, how similar are they to each other? Like, is this guy similar to this guy right here? Well, we see that they're both green, but does that mean that they're the same? So we need some way to kind of categorize these things. And this is where the next scientist kind of came into play. Carlos Linnaeus, right, started to develop this taxonomic system for organizing these organisms, whether they be plant or animal, or in our case, microorganisms, into different groups based upon, in his case, physical characteristics, right? And he came up with 6 different categories. So bacteria, right, archaea, fungi, protozoa, algae, and then small multicellular organisms, right? Some of these are kind of classified as parasites, and we'll see why they're classified in there and why we use them in that classification scheme. So just going a little bit more detail to each one of these. When we first look at the first ones, and these are the prokaryotes. Prokaryotes have this characteristic of generally being single cellular. So they're unicellular. They only have one cell. The whole organism is just one cell, and they don't have a true nucleus, right? So they may have a nucleolus, but they don't have a membrane bound nucleus like you would find in a eukaryotic cell. They're also pretty small, right? And they're found essentially everywhere on this planet. There is an environment that can support some type of microbial life pretty much everywhere on this planet. right? So when we look at them, they're found in a lot of diverse areas that usually have moisture associated with them. Some of them have the ability to actually reproduce. Some of them have the ability to reproduce in areas that you wouldn't really consider areas that would be hospitable to normal organisms. Like it may be a hot spring that is extremely hot, right? And you might find some archaea living in those areas. So the characteristics of these organisms, like these guys that don't have peptidoglycan and may have polymers that make up their cell wall, may afford them the ability to survive in areas where you normally wouldn't be able to find life. Right, and bacteria do have peptidoglycan in their cell walls, and some don't even have cell walls at all. When you look at like the mycobacteria, right? So moving on, right, we see some cells here. Now here's an example of two different types of cells. Here is a cheek cell. So this is like a human eukaryotic cheek cell. So if you looked inside your cheeks, right, and scrape some cells out and stain them and put them on a microscope slide, this is kind of what they would look like. Now, if you see all these little dots around them, these little dots all around them are prokaryotic cells. These are bacteria cells. So you can notice the huge disparity in size. Like their cells are so much smaller than your cells. Right, now, the next type is the fungi, right? Fungi, you may be familiar with stuff like mold or single cell yeast. They do have ways of reproducing that are a bit interesting. Some of them can do sexual reproduction, while some do asexual reproduction. They can do budding. They can do sexual spores. They have all these different ways to reproduce, but these are eukaryotic cells like you. So that eukaryote means true nucleus. So they have a membrane-bound nucleus just like you, and they do have cell walls. Like when we look at bacteria cells, they do have cell walls like them, and as well as plants have those as well. Right, and here you can see some of those spores. Right, these are the things that when you kick a mushroom and you see a little puff of smoke, it actually isn't smoke, it's actually spores, it's reproductive spores, so you help them reproduce doing that. Right, now here you can see this little bud coming off of this cell, and this little bud will be an identical copy to this individual here, so that will be asexual reproduction. Now, moving on to the next one, and these guys are pretty interesting. These are eukaryotes, but they're single-celled eukaryotes. So these are little small guys, but they operate a lot like animals. They act like animals, right? They have the same kind of requirements of nutrients as animals. Most reproduce asexually, but some can actually do sexual reproduction. Now, they're most noted for their means of locomotion, so their ability to get around. And they have three primary ways that they get around. Pay attention to this. The first one is pseudopodia. Pseudo meaning false, pod meaning foot. So false foot. This false foot allows them to move around in their environment, right? The next one is cilia. With cilia, they have these short little projections that come over and sometimes over the entire cell that allow them to act like little boat oars and row and move around their environment. The last is flagella. You may be familiar with flagella because things like sperm cells have flagella as well. Bacteria can have flagella as well. But these are like a whip-like tail that are longer than cilia, right, which allow them to propel themselves through their environment. So 3 different modes of locomotion: pseudopodia, cilia, and flagella, right? So remember, protozoa are known for their 3 different modes of locomotion: pseudopodia, cilia, and flagella. Then we move on to algae. They can be both uni or multicellular. They are photosynthetic. So this means they actually use carbon dioxide and water and energy from the sun in order to produce their own sugars, which go to build their structure and provide them with an energy source in order to live. Right now, when we characterize them based upon 2 different components: what pigment they use in order to gather the energy from the sun, and then the composition of that cell wall. Right? And here you can see some examples of algae. In fact, some of my favorite ones, like diatoms, they come in all different shapes and varieties. And actually, they almost look like art when you look at them under a microscope. You might have a chance to look at that in the lab later in the semester, depending on what goes on with this current situation. right? And then finally, we have some of these microbes that are classified as such because of how they interact with us. Things like parasites and viruses. So when we look at parasites, parasites do have forms within their developmental stages where they are microscopic and it can be viewed with a microscope. This is why they're classified as microbes, right? And viruses are microscopic entities, even though they aren't considered alive per se. They do interact with us on that microscopic scale. This being a bacteria cell, and these guys being viruses for bacteria. Is what we call the bacteriophage, are infecting this bacteria cell, and then they're being assembled inside. And then, depending on the type of virus, either going to have that cell lyse and burst open or incorporate that genome into the host genome, and then it kind of stays around, right? We'll learn about those as we go along in the class as well. So early on, there was a lot of back and forth about how life actually came into existence, and in particular, in our case, microbial life. The common thought was that spontaneous generation was a thing. So from nothing, you could develop these microbes and they can cause all different types of problems because they just come out of kind of thin air. Um, Now, how we got to the answer of how microbes come into existence kind of came from some other things like fermentation. When we look at fermentation, we may be thinking about making like alcohol, and this is one form of fermentation. And from experiments done by Pasteur in fermentation, we actually figured that out, and it kind of gave us more insight into what actually does cause disease and help us to develop what we call germ theory, that a lot of the diseases that we have actually come from microbes. So if we know that these diseases are coming from microbes, then can we use things that prevent the microbe from replicating in order to curb that disease, right? So this goes into preventative methods, and this is what we now know, right? So early on in kind of the history of microbiology, it was always thought that people could philosophize and come up with answers to problems. And there's a problem with that. An idea doesn't really hold weight unless you have a way to test it. So at this point, when Aristotle was around and he had this thought of abiogenesis, this was kind of the prevailing thought that life could come from no life, right? But it wasn't until scientists came along that kind of challenged that idea, and challenged that idea with not just another philosophy, but with evidence coming from experiments, that it started to kind of push the needle in the direction of things like germ theory and understanding that microbes don't come from nothing. They actually do come from other microbes, right? So one of the first experiments that was crucial in this was the experiment done by Redi. So Redi was a Italian physician who kind of challenged that whole thought of abiogenesis. And the way he did that is by using meat, right? What he noticed is that maggots always tend to form on meat. So the common thought would be that maggots come from meat, right? Well, he said, well, if maggots really come from meat, if I change up the scenario that this meat is in, then the maggots should still come out of the meat. So what he did is he had multiple flasks, right? Some flasks unsealed and some flasks sealed. Some flasks would actually gauze over the top. And what he did is he allowed it to just sit there, right? Now, the maggots that had the unsealed flask, the meat that had the unsealed flask had flies to land on it. And then eventually maggots showed up. The one that was sealed never developed any maggots on top of it. And in fact, the one with the gauze, which allowed essentially the smell of the meat to radiate out into the environment, but didn't allow the flies to land on the meat, actually had the flies landing on the gauze, right? And laying their larvae on the gauze, right? Instead of the meat. So what is this showing us? That the maggots aren't actually coming from the meat. The maggots are coming from the flies, right? But this alone wasn't enough to kind of nail down that spontaneous generation wasn't a thing. Right? So we needed additional scientists to come along and kind of help us to figure this out. So one of the scientists that helped us to figure this out, and I said, well, not really figure it out, but helped us to give some insight. And this was Needham. So Needham took some beef gravy infusions along with plant infusions, and he boiled them and then sealed them tightly. Now, what he saw was that eventually it formed like this cloudy mess, right? And this cloudy mess, he concluded that there's a life force that existed that it can cause like inanimate objects to become alive. Right now, there were scientists that came after him and said, "No, that's not right. Maybe you just didn't boil it enough to kill all the microbes that may have been in there." Right. And then we know now that this is the case. He just simply didn't boil it enough in order to kill all the microbes in there. But he counted, well, if you boil it too much, then you kill the life force. You need a life force in order to make sure that these things can actually come out of nothing. Right. So Needham kind of gave some contradictory information to some of the experiments that were going on at the time. So what do? Well, you have more experiments. So another experimenter came along and said, well, Needham didn't really do due diligence with his experiments. And this is Spallanzani, right? He said, well, he just simply didn't heat the valves enough in order to kill all the microbes, or he just didn't seal them tight enough to keep out all the microbes. So the air has microbes, is what he claimed. And if the air can get into that beef infusion, then it can contaminate it, right? Well, people criticized his work and they said, well, hey, if you heat it up too much, you do destroy that life force. So you can't heat it up for that long. The life force is gone. It won't be able to just spontaneously generate these organisms. And in fact, you need air. You got to have air in order to get in. So along came a guy named Louis Pasteur. You may be familiar with him through this whole pasteurization process. You see it all over milk and juices and things of that sort. And he wanted to kind of nail down and kind of put this whole thing to rest of what actually cause spoilage. Right now, his work was supported in the future by vintners, so people making wine. But initially, he was just a really hard and dedicated worker, so much so that his wife wanted to spend time with him. And he said essentially to her, leave me alone. I'm going to make your name famous. Right. And for all practical purposes, he did. Now, he was an extremely hard worker. He didn't have many space. I believe he worked under his staircase with these experiments. And he did some very interesting experiments with a particular type of flask. This is a swan neck flask. Now, what's so important about this swan neck flask is the swan neck flask will allow air to get in, but because microbes don't fly, they can't actually get in. The air can flow through, but the microbes themselves can't get in, which means that the life force can get into the infusions, but the microbes cannot get into infusions. So let's kind of look at what he did. So here you have this infusions, but now you have this swan neck flask, right? So you boil the infusions to make sure that everything in here is killed. So once everything is killed, air can still get in and air can still get out, right? So air can flow through, but when those microbes come in, they settle right here in this little bottom of this U-tube. So this can stay for very long periods of time and remain sterile because the microbes can't get up, even though air can get up. So this kind of puts to rest that whole life from no life thing, where it says, well, no, we need some life. Now, he took it a bit further where he cracked this and took some of these microbes on this dust. This looks like dust to him, and from this area, but had microbes in it, and he put it into that infusion. And what did he find? Well, what he found was that it started to get contaminated and start to get cloudy, what we call turbidity, because these are the things that were actually causing the spoilage of that infusion, right? So taking a little bit further, right, we need to now figure out, well, if this is causing spoilage, then what causes other things? Like what causes wine to be made? If I want to make wine, what do I need? Right? Do I need this microbe, that microbe? Is it yeast? Is it bacteria? What's causing my wine to spoil? So all of these questions kind of help to develop, and this whole process of determining whether spontaneous generation was a thing helped to develop the scientific method. Right now, the basics to the scientific method is once you make an observation about your world, then you make a hypothesis that helps you to figure out the answer to that observation, right? So once you have this hypothesis you test it with a controlled experiment. Either it's going to be proven correct or be disproven based upon the data you collect. If it's proven correct, you accept it. And eventually, with a mass of information, it can lead to things like theories and laws, right? If it's disproven, you reject it and you can modify that hypothesis and retest it again. So it's pretty basic. We all do it. Let's imagine we had a situation where we went out to the car in the morning and we tried to turn it on, right? And we realized that the car did not turn on. Well, we just made an observation, right? And then very quickly, we don't even think about this, but very quickly, our mind starts to go to creating a hypothesis from that question. And what do we probably do? Oh, it's the battery, right? So we think it's the battery. So what do? Right? Well, we may go in there, clean the connectors to the battery. And let's say we clean the connectors to that battery, right? And then we observe that the car turns on. Well, if the car turns on, that experimental data supports the hypothesis that we just made. So we accept it. And eventually, right, not in this case, because this is just a very small scale experiment, but if we see this over and over again, we can accept it as a theory. So if we hear particular sounds, we know that it may be the battery. Now, if we do this same thing and say it's the battery, but it doesn't start, even though we may put a new battery in there through our experiment, then we have to reject that hypothesis. We can modify it and say, hey, maybe it's not the battery. Maybe it's the starter, right? Maybe it's something else. So we project the hypothesis and create a new one and then retest again. So this is the basics to the scientific method. You should have seen this by now. You should be well accustomed with this. So the wine industry essentially wanted to put them to work. They wanted to figure out what was causing their wine to spoil and what could cause them to make higher yield alcohol from the grapes that they were producing. Right now, some people would say, well, it's the air. The air causes the fermentation process to happen. So you need air in order for the ferment. Well, we need to figure out with certainty what it is so that we can have this high yield of alcohol, because that's what you want to go for. So what did he do? Well, he did multiple experiments to basically nail down what was the causative agent to fermentation. Like, what caused fermentation? Was it yeast? Was it bacteria? Was it air? Right? So now here's his experiments here. So we had some fermented grape juice, right? And he noticed that there were some microbes within that fermented grape juice. And what he saw was there was both yeast and bacteria. So if these are your two prime suspects, you want to kind of isolate them. Say, let's get just the yeast. Let's get just the bacteria alone. And then put them into some grape juice and see what happens. So the first one, he takes the grape juice, heats it up until it kills all of the microbes in it, closes it off, and just leaves it. So if air is the thing that's fermenting, right, then it should just ferment because there was air in there, but there's no air. So if the juice remains free of microbes, that means life can't come from anything. Spontaneous generation is essentially not a thing. So if air is the thing that's doing it, if we use that swan neck flask again, it should ferment it, but it never does because those microbes that stay in the bottom of that little YouTube can't get up in there into there to cause fermentation to happen. Right? Then he heats up another flask and he introduces bacteria. We call it inoculating bacteria into that flask. And what happens is it produces acid. Acid is not what you're going for, right? So bacteria is not causing that fermentation process that leads to alcohol. It's causing a fermentation process that leads to acid. We call it acid fermentation, right? Lactic acid fermentation in particular, right? Now, this is acetic acid fermentation, right? So now you do the same thing with yeast. You heat up to kill all the microbes that's in there. You inoculate it with yeast. And what do you have? You have alcohol. So what did he just figure out? Well, he figured out that fermentation, in order to produce alcohol, requires yeast, right? So yeast is what you need in order to make that alcohol. Okay? Now, he also came up with the pasteurization process, obviously, right? We passed the war, pasteurization, and it's a process of just heating up liquids to kill most of the bacteria that may cause disease or spoilage, right? So that these things last longer, right? So very important when it comes to industrial sense and actually keeping our food supply there. But now we know that microbes can produce a lot of products for us, everything from cheese to breads to vitamins to antibiotics to a lot of the hormones that we take. Right. So microbes have become extremely useful, especially with our knowledge of genetics. As we start to learn how genetics works and how microbial genetics works, we can start to manipulate these guys in order to do things that we want them to do. Now, the next scientist that came along was Buckner. Now, Buckner had this question of, does fermentation require yeast? Does it require the actual living cell in order to ferment? Well, the quick answer is no, right? All it requires is all of the enzymatic reactions and thereby the enzymes that allow for that production of alcohol to proceed. So you just have to have them in the right combinations at the right time, and you can definitely do it. So what did Buckner bring into existence? Essentially, biochemistry. He started to understand how enzymes and metabolism works in order to actually still produce the products that we're going for, but in a cell-free environment. So there's no living things that's necessary in order for fermentation to go forth. It's just all of the biochemical reactions necessary in order for fermentation to happen. Right? So now that we have this idea that fermentation can be caused by yeast, well, if it can cause fermentation, what else could it cause? Could it cause disease? Right? So thinking about that kind of helps us develop this whole germ theory that a lot of the diseases that we are accustomed to are caused by germs, right? Microbes. So these pathogens are the things that are causing diseases. Like if I asked you, what causes AIDS? Well, most people say, well, HIV, right? HIV leads to the condition that we know as AIDS. But how would you figure that out? Like, how would you figure that out if someone didn't tell you? Like, what would we do? Well, this is what this next guy, Koch, was actually kind of concerned with. How do we figure this stuff out? Right now, Koch did a tremendous amount of work. But some of his most important things was coming up with these postulates, right? We call them Koch's postulates that helps us to determine the etiology or the causative agent to disease. Now, he did work with anthrax, right? Because he wanted to figure out what bacteria causes anthrax. Right? So this is pretty simple, right? It would seem so. But remember, when you don't have all this background information and you're developing this yourself, it is very, very difficult. So in order to figure this stuff out, he had to develop a lot of techniques. So he developed staining techniques, simple staining techniques where you can actually start to see these microscopes. Seeing the microscopes was very important to help to determine what you may be looking at. You may be familiar with other types of things, like maybe the gram stainings that can help us to determine the difference between gram negative and gram positive bacteria. So understanding what you have is important. Also, being able to grow and culture these things. If I take a sample from an individual, I need to know what they are, but in order to know what they are, I got to have enough of them to where I can see them. So he actually developed petri dishes, right? Ways to actually determine what bacteria we were looking at, to transfer bacteria from one area to another using the wire loop transfer system, right? So all of these were very important in helping to determine what bacteria we were actually dealing with. Now, if you look here, you see a lot of different types of bacteria. You can see they are different, right? Each shape, right? If you look at the border of some of the borders jagged, while other borders are very smooth. Some of them kind of pinkish, some of them kind of reddish, some of them kind of yellowish. All these give you an indication that you're looking at a different microbe, right? So what do these postulates actually say, right? What these postulates basically say is, if you believe that a certain microbe is the causative agent, it has to first be found in all of the individuals that have that disease, right? And it can't be found in individuals that don't have that disease. Right? You then have to take that agent away from that individual. You have to be able to remove some of that agent from that individual and isolate it, right? You should then be able to take that isolate and inoculate a healthy individual with it, and they have to get that same disease and have those same symptoms as that first individual who you derived it from, right? And it should be found in all individuals. So these are his postulates in a very kind of succinct way, but this is what he does. So you should be able to, one, right, see that same agent in all individuals that have that disease. You should be able to isolate that causative agent, right, and then inoculate it into a new healthy individual, right, and that healthy individual should have the same symptoms as the first individual and develop that disease, right? So these are Koch's postulates. All right. So moving on, right, we have some other individuals that were pretty important in the kind of the history of microbiology, and one of them is Gram. So Gram, you're probably familiar with, I just mentioned Gram staining. This is one of the first steps that you'll do in order to figure out what you're dealing with. So you have Gram positive and Gram negative bacteria. Gram negative being kind of the more dirty and dangerous of the two, right? The Gram negatives are pink. They stain pink. And the Gram positives stain purple. Gram positives, purple. Gram negatives, pink. So positively purple, right? Now, the next individuals are all very important, but they more went so into helping prevent infection and disease. Some of the stories are just horrible. Like the first individual on this list, Semmelweis, he was worked in obstetrics in Vienna. So it was at a teaching school where he saw a very high death rate in both maternal and infant mortality. He also saw individuals like a doctor get cut and died from what they call perpetual fever, which is essentially just streptococcus. He had a fever from streptococcus. So what he kind of championed was hand washing, and hand washing what we call chlorinated lime water. Now, the people at his hospital, that teaching hospital, basically shunned him. They said, this is stupid. There's no reason to do this. Why would I do this? Now, even with the data that he got when he actually kind of enacted this whole hand washing with the chlorinated lime water showed that the death rate dropped tremendously. They still didn't accept him in the whole of Europe. So he kind of went back to his hometown in Hungary. And unfortunately for him, he actually died of a streptococcus infection, the same thing that he was kind of going to champion against, and just using normal hand washing process is the same thing that he died for.
Years before the germ theory of disease was developed, the Viennese physician Ignaz Semmelweis came to the startling discovery that doctors could save the lives of patients simply by washing their hands. Although he couldn't explain why hand disinfection was so effective, his research in 1847 would go on to transform the way surgery is carried out and infectious diseases are controlled today. Known locally as the savior of women, Semmelweis initiated hand-washing policies which helped drastically reduce the mortality rate from childbed fever in the Viennese obstetrics clinics where he worked. Childbed fever, or puerperal fever, is the infection of the female reproductive organs following childbirth and was a common cause of death at the time. Semmelweis's attention was first drawn to the mortality rate of the clinics he worked at, because women would beg to be admitted to the clinics run by midwives rather than those staffed by students. The women were so desperate to avoid the student clinics that they would rather give birth in the street. After looking into the mortality rate of the two clinics, he found that the student-run clinic did indeed have a much higher mortality rate from puerperal fever, sometimes 3 times higher. After eliminating various factors, Semmelweis came to the conclusion that the students carried something from the dissection rooms where they performed autopsies to the patients they examined during labor. He ordered the students to wash their hands in a solution of chlorinated lime before each examination. The mortality rate fell from 18% to 1%. Despite the success of his policy, Semmelweis faced the wrath of the medical community. He could offer no acceptable explanation for his findings, as the notion that there were germs causing disease was unaccepted, and various doctors were offended by the insinuation that their hands were dirty. He was ostracized and ridiculed by the leading medical figures of his time and eventually driven out of Vienna. After struggling for years to promote his hand disinfection policies, Semmelweis was admitted to an insane asylum at the age of 47. He died 14 days later.
Then you have individuals like Lipster, which actually championed antisepsis. So he sprayed wounds with carbolic acid, which is what we'll eventually learn as a phenol, which helps to reduce the odor and decay and stuff like sewage. Right now, how does it do that? It retards microbial growth. That smell is coming from microbes breaking down materials in order to eat, right? And then you have individuals like Florence Nightingale. You're probably familiar with her, being that most of you are probably trying to go into nursing. She was a nurse within the Crimean War who was an advocate for cleanliness and aseptic techniques. She advocated for cleaning dressings and clothes. And she was a big advocate for nursing education, educating the nurses in these different techniques in order to keep their patients alive. Right. Other individuals like Snow were very important in the cholera outbreak in the water supply. So he kind of championed a couple of fields into existence, one being infection control and the other epidemiology. Once you find out, like through epidemiological studies, that this disease is being transmitted because something is happening in this particular area, then you can start with that whole infection control. Right. Then Jenner kind of brought the modern vaccine into existence by infecting individuals with cowpox in order to protect them from smallpox. Kind of an interesting story. I'll kind of cut away to a little video. So.
10,000 years ago, a deadly virus arose in northeastern Africa. The virus spread through the air, attacking the skin cells, bone marrow, spleen, and lymph nodes of its victims. The unlucky infected developed fevers, vomiting, and rashes. 30% of infected people died during the second week of infection. Survivors bore scars and scabs for the rest of their lives. Smallpox had arrived. In 1350 BC, the first smallpox epidemics hit during the Egypt-Hittite War. Egyptian prisoners spread smallpox to the Hittites, which killed their king and devastated his civilization. Insidiously, smallpox made its way around the world via Egyptian merchants, then through the Arab world with the Crusades, and all the way to the Americas with the Spanish and Portuguese conquests. Since then, it has killed billions of people, with an estimated 300 to 500 million people killed in the 20th century alone. But smallpox is not unbeatable. In fact, the fall of smallpox started long before modern medicine. It began all the way back in 1022 AD. According to a small book called The Correct Treatment of Smallpox, a Buddhist nun living in a famous mountain named Ome Shan in the southern province of Sichuan would grind up smallpox scabs and blow the powder into nostrils of healthy people. She did this after noticing that those who managed to survive smallpox never got it again, and her odd treatment worked. The procedure, called variolation, slowly evolved, and by the 1700s, doctors were taking material from sores and putting them into healthy people through 4 or 5 scratches on the arm. This worked pretty well, as inoculated people would not get reinfected, but it wasn't foolproof. Up to 3% of people would still die after being exposed to the pus. It wasn't until English physician Edward Jenner noticed something interesting about dairy maids that we got our modern solution. At age 13, while Jenner was apprenticed to a country surgeon and apothecary in Sodbury, near Bristol, he heard a dairymaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pockmarked face." Cowpox is a skin disease that resembles smallpox and infects cows. Later on, as a physician, he realized that she was right. Women who got cowpox didn't develop the deadly smallpox. Smallpox and cowpox viruses are from the same family, but when a virus infects an unfamiliar host, in this case cowpox infecting a human, it is less virulent. So Jenner decided to test whether the cowpox virus could be used to protect against smallpox. In May 1796, Jenner found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hand and arm, caught from the udders of a cow named Blossom. Using matter from her pustules, he inoculated James Phipps, the 8-year-old son of his gardener. After a few days of fever and discomfort, the boy seemed to recover. 2 months later, Jenner inoculated the boy again, this time with matter from a fresh smallpox lesion. No disease developed, and Jenner concluded that protection was complete. His plan had worked. Jenner later used the cowpox virus in several other people and challenged them repeatedly with smallpox, proving that they were immune to the disease. With this procedure, Jenner invented the smallpox vaccination. Unlike variolation, which used actual smallpox virus to try to protect people, vaccination used the far less dangerous cowpox virus. The medical establishment, cautious then as now, deliberated at length over his findings before accepting them. But eventually, vaccination was gradually accepted, and variolation became prohibited in England in 1840. After large vaccination campaigns throughout the 19th and 20th centuries, the World Health Organization certified smallpox's eradication in 1979. Jenner is forever remembered as the father of immunology. But let's not forget the dairymaid Sarah Nelms, Blossom the cow, and James Phipps, all heroes in this great adventure of vaccination, who helped eradicate smallpox.
You can see how this worked. Right now, there's Ehrlich. Ehrlich championed the field of chemotherapy. He wanted what's called a magic bullet. He wanted something to be created that would essentially kill everything. Right, it would take care of every disease, and he actually used arsenic to treat syphilis. Now, will arsenic kill the little spiral bacteria that causes syphilis? Absolutely, but unfortunately, arsenic could also kill you as well. So you always have to consider that, and we'll get to eventually to a term where we talk about selective toxicity, which you want it to kill the organism that's causing the problem while leaving the host as unaffected as possible. Right. So we'll kind of stop here, right? And we'll continue on the second half of this lecture a little bit later when we do our live lectures.